Imagine for a moment a world where your own body isn't just, hosting the medicine, but is the medicine, a medical breakthrough that sounds, well, less like a prescription, more like science fiction maybe. But it's very real. We're talking about a living drug using a patient's own cells, re-engineered, sort of supercharged, to fight off some really tough diseases. Today, in this deep dive, we're really getting into one of the most exciting advancements around, CAR T-cell therapy. Everything we'll discuss comes from the articles, the research, the notes you've shared with us, and our mission to pull out the key bits of knowledge. We want to understand its potential, how it works, like down at the molecular level, where it's being used now, and also, importantly, the challenges and what's next. We'll try to make this complex stuff easy to follow and, honestly, pretty fascinating.
Absolutely, and it is genuinely innovative. It flips the script on cancer treatment. Instead of just external drugs, we're harnessing the body's own immune system, turning it into a really targeted weapon. It's this amazing mix of biology and, well, engineering.
Okay, so let's start right there. The core idea. CRT cell therapy, it's a form of immunotherapy, right? It takes a patient's T cells, those are white blood cells, your body's security force, basically, and modifies them genetically. The aim is to train these T cells to specifically find and then attack the cancer cells.
And the genetic modification part is really the key,
Yeah.
That's why it matters so much. We're not just giving the immune system a temporary nudge. We're fundamentally rewriting the instructions for these T cells. It's like turning general soldiers into, I don't know, precision-guided specialists designed to hunt down cancer with incredible accuracy.
And it's already shown some, well, credible success, especially with certain blood cancers, right? Like specific leukemias, lymphomas.
Exactly. That's where the big breakthroughs have been so far.
Though research is definitely happening for solid tumors too, which I imagine is a whole different ballgame.
Oh, absolutely. Solid tumors present unique hurdles.
Okay, let's unpack this. To really get it, we need to follow the journey of this living drug. It starts, maybe surprisingly, with just drawing blood. Step 1, T-cell collection. A leukapheresis, they call it. So they draw blood, spin out the T cells, and return the rest to the patient. Kind of like donating plasma.
Very similar process, yes. Highly specialized, but the principle is the same. Collect the specific cells needed.
Okay, so you've got the T cells. Then they go to the lab. Step 2, T-cell engineering. This is the modification part.
Precisely. This is where the magic, or rather the science, happens. In the lab, these T cells are engineered to express CAR's chimeric antigen receptors on their surface. Think of CARs as custom-built sensors, synthetic proteins designed to lock on to specific markers or antigens on the cancer cells.
Like CD19, I read that's common in B-cell leukemia.
Exactly like CD19. It's a well-known target. And they usually use viral vectors, think of them as like disarmed viruses acting as delivery vehicles to get the gene for that CRR into the T cell's DNA.
Viral vectors. Sounds intense, but it's a standard technique in gene therapy, right?
A very established and refined technique, yes. It's how you precisely insert the new genetic code.
Okay, so now we have engineered CAR T cells, but not enough of them yet. So, step 3, T-cell expansion.
Correct. You need an army, not just a few soldiers. These modified cells are grown in the lab, multiplied into large numbers. This takes usually 1 to 2 weeks. You need billions of these cells for the therapy to be effective.
Billions. Wow, okay. Before they go back in, there's another step. Conditioning chemotherapy. Lymphodepletion.
Yes, step 4. Before the CAR T infusion, the patient gets a round of chemotherapy. Now, the main goal here isn't necessarily to kill the cancer, but to reduce the patient's existing immune cells.
Why do that? Seems counterintuitive.
It creates space, you see. It makes the body environment more receptive for the new CAR T cells. Less competition, better signals for them to grow and persist. It helps them engraft properly.
Okay. Clearing away. Makes sense. And then finally, step 5, CRT cell infusion.
That's it, the big moment. The engineered expanded CAR T cells are infused back into the patient intravenously, like a standard infusion. Once they're back in the bloodstream, they start their search and destroy mission. They seek out cells with that target antigen, bind to them, and, well, eliminate them.
It's quite a journey for those cells.
It really is. And what's fascinating here is just how personalized it all is. You're essentially creating a custom-made living drug for each individual. It's this incredible blend of the patient's own biology and really sophisticated lab work. Very complex, very precise.
So how do they actually work once they find the target, the mechanism? You mentioned CARs, these receptors. This is where it gets really interesting, right? The molecular details.
Absolutely. Let's zoom in on the CIR itself.
Okay. You said 3 main parts.
Right. First, the outside part, the extracellular domain. This is the bit that sticks out from the T cell. It's often derived from an antibody fragment, and its job is to recognize and bind to that specific antigen on the cancer cell. It's the targeting system.
Like the lock and key mechanism.
Exactly. Then there's the transmembrane domain. Pretty simple. This just anchors the whole CR structure firmly in the T cell's membrane, holds it in place.
Okay, anchor. Got it. And the third part, inside the cell.
That's the intracellular signaling domains. These are crucial. When the outside part binds to the antigen, these inside parts act like switches. They transmit the signal into the T cell, telling it, target locked, activate.
And these switches have gotten better over time, I understand. Generations of CARs.
Yes, huge improvements. First generation CARs only had one main activation switch called CD3 zeta. It worked, but the T cells didn't stick around for long or multiply very well. Second and third generation CARs, the ones mostly used, now added extra signaling domains, co-stimulatory domains like CD28 or 41BB.
Co-stimulatory, like giving it an extra boost.
Precisely. Think of CD3 zeta as the on switch, and these co-stimulatory domains as the accelerator and staying power signals. They tell the T cell not just to activate, but to proliferate, make more copies of itself, and to survive longer in the body. Much more potent and durable response.
So the swayer binds the target, the switches flip inside.
And the T cell goes into action. It directly kills the cancer cell it's bound to. It starts dividing rapidly, creating more CAR T cells right there at the site, and it releases cytokine signaling molecules that basically call in reinforcements from other parts of the immune system. It orchestrates a whole attack.
A really powerful, targeted immune storm.
When it works well, yes. It leverages the T cell's natural killing ability, but gives it that crucial specificity.
So what does this all mean for patients? We've talked about the science, but the real-world impact. That's what matters.
And there are actual approved therapies now, right? This isn't just theory anymore.
Not at all. As of 2024, several CAR-T therapies are FDA approved and changing lives. It's remarkable progress.
Can you give us some examples?
Sure. You have therapies like Kymriah and Yescarta. Both target that CD19 antigen we mentioned. Kymriah is used for certain types of B-cell ALL, acute lymphoblastic leukemia, and also diffuse large B-cell lymphoma, DLBCL. Yescarta covers DLBCL, primary mediastinal B-cell lymphoma, follicular lymphoma, several B-cell cancers. Okay, so CD19 is a big target for lymphomas and leukemia.
A very important one. Then there's Tecartus, also CD19, approved for mantle cell lymphoma and AL. And Breyanzi, another CD19 targeted therapy for large B-cell lymphoma.
So lots focused on CD19. What about other cancers?
Multiple myeloma has seen huge progress too. There, the target is usually BCMA, B-cell maturation antigen. Two key approved therapies are Abecma and Carvykti. Both target BCMA and have shown really impressive results in patients with relapsed or refractory multiple myeloma.
That's fantastic news for myeloma patients. These approvals, especially for cancers that were maybe resistant to other treatments, that's significant.
It's monumental.
Yeah.
Many of these patients had run out of options. CAR-T offers a completely new mechanism, and for some, it leads to deep, durable remissions. And it raises an important question, why these targets? Why CD19 and BCMA?
Right, what makes them good targets?
Well, ideally, a target antigen should be highly expressed on the cancer cells, but have limited expression on essential normal tissues. CD19 and BCMA fit that bill pretty well for B-cell cancers and myeloma, respectively. They are consistently found on the malignant cells. This allows the CRT cells to be very specific, minimizing damage to other healthy parts of the body, although, as we'll discuss, it's not always perfect.
Okay, so high specificity is key, but it sounds like it's not always smooth sailing. Despite the power, there are significant considerations, right? Side effects.
Absolutely. It's a very potent therapy, and that potency can come with serious, sometimes life-threatening side effects. We have to be upfront about that.
The big one you hear about is cytokine release syndrome, CRS.
Yes, CRS is probably the most common serious side effect. It happens when the activated CRT cells rapidly kill cancer cells and release a flood of cytokines. Remember those signaling molecules. Too many at once can cause systemic inflammation. Symptoms range from fever and flu-like symptoms to dangerously low blood pressure, difficulty breathing, organ dysfunction. It needs immediate management.
How do they manage it?
We've gotten much better at it. Drugs like tocilizumab, which blocks a specific cytokine receptor, IL-6 receptor, are very effective. Corticosteroids are also used. Close monitoring in specialized centers is critical.
Okay. What about neurotoxicity, ICANS?
Right, ICANS, immune effector cell-associated neurotoxicity syndrome. This is another significant concern. Patients can experience confusion, delirium, difficulty speaking, seizures, even brain swelling in severe cases. The exact cause isn't fully understood, but it seems related to the inflammatory environment created by the CAR T cells, maybe cytokines crossing into the brain.
That sounds scary. Is it manageable too?
It often is, yes, with supportive care and sometimes corticosteroids, but again, it requires expert management and monitoring. It usually resolves, but it can be serious.
Any other major side effects?
Well, there's B-cell aplasia. If you target CD19, you wipe out the cancerous B cells, but you also wipe out the healthy B cells because they also have CD19.
Ah, so that impacts the immune system long term.
It does. Patients lose the ability to produce antibodies effectively, making them prone to infections. This is managed with regular infusions of intravenous immunoglobulins, IVIG, essentially giving them antibodies from donors.
Okay. And tumor lysis syndrome.
Yes. If the therapy works very quickly and kills a large amount of tumor cells rapidly, the content spilling out can overwhelm the kidneys and disrupt electrolyte balance. Requires careful monitoring and preventative measures.
So significant potential side effects. And what about other limitations, broader challenges?
There are definitely hurdles. Cost is a huge one. These are personalized therapies, complex to manufacture. The price tag is often hundreds of thousands of dollars per patient. Accessibility is a major issue.
That's a massive barrier.
It is. Then there's the manufacturing time. It takes weeks to collect the cells, engineer them, expand them, get them back to the patient. For someone with very aggressive cancer, that delay can be critical.
Weeks. Yeah, that's a long time to wait.
Also, not all cancers respond. It's been most successful in these blood cancers. Solid tumors are much harder.
Why are solid tumors tougher?
Several reasons. Getting the T cells into the tumor mass can be difficult. The tumor microenvironment itself is often immunosuppressive, actively shutting down T cells. And finding good, unique targets on solid tumors that aren't also on vital organs is tricky.
More complex targets, hostile environment.
Okay. And even in blood cancers where it works well, relapse can happen. Sometimes the cancer cells adapt. They might stop expressing the target antigen, like CD19, becoming invisible to the CAR T cells. This is called antigen escape.
The cancer evolves to evade the treatment.
Exactly. It's a constant battle. So it would connect us to the bigger picture of personalized medicine. These challenges, the side effects, the cost, the manufacturing, the resistance, they're all major focus areas for current research. Balancing the power with the risks, making it broader, safer, more accessible, that's the goal.
Right. And that perfectly leads us to the future. Where is CAR T heading? What are the exciting advancements?
Oh, the innovation is moving incredibly fast. It's a really dynamic field.
One thing I've heard about is off-the-shelf CAR AT allogeneic.
Yes, allogeneic CAR T. This is a huge area of research. Instead of using the patient's own T cells, the idea is to use T cells from healthy donors. You could potentially create large batches of CAR T cells in advance, store them, and have them ready to go immediately when a patient needs them.
That would solve the manufacturing time issue and maybe the cost.
Potentially both, yes. It could make the therapy much faster and more widely accessible. The big challenge there is preventing the donor T cells from attacking the patient's body graft versus host disease and preventing the patient's immune system from rejecting the donor CAR T cells. Clever gene editing techniques are being developed to try and overcome this.
Okay, fascinating. What else is on the horizon?
Multi-antigen CARs to combat that antigen escape problem we talked about.
Targeting more than one thing at once.
Exactly. Design CARs that recognize 2 or even more different antigens on the cancer cell. That makes it much harder for the cancer to hide just by losing one target.
Like having multiple locks on the door?
A good analogy, yes. Another approach is logic-gated CARs. These are smarter CARs. They might be engineered to only activate if they detect, say, 2 specific antigens present, increasing specificity, or maybe they only activate strongly within the tumor microenvironment, reducing side effects elsewhere, adding layers of control.
More precision, more safety features built in.
Right. And the field is looking beyond T cells, too. CAR-NK therapies using natural killer cells, which might have a better safety profile, or CAR macrophages using immune cells that are good at infiltrating solid tumors. Engineering different types of immune cells opens up new possibilities.
Wow, so it's expanding beyond just T cells.
Definitely.
Yeah.
The core concept of engineering immune cells with chimeric receptors is being applied much more broadly now. The relentless innovation here is really focused on overcoming those current limitations we discussed, making CAR-T safer, making it work for solid tumors, beating resistance, making it accessible. The potential to transform how we treat not just cancer, but maybe other diseases too, is immense.
Okay, so let's try and wrap this up. CAR T-cell therapy. It's truly cutting edge. A personalized cancer treatment using the patient's own immune system, specifically their T cells, reprogrammed to find and fight cancer. It's complex, no doubt. It's
Hmm.
expensive right now, and it carries real risks, serious side effects. But the results can be incredible, life-saving even. And the research is pushing forward so fast, tackling those challenges head on.
It really is a paradigm shift. And as we get better at, re-engineering our own cells, our own biology, it really makes you think. As we delve deeper into reprogramming these cellular mechanisms, it prompts us to wonder what other complex diseases, maybe autoimmune disorders, infectious diseases, might someday be redefined or even overcome by unleashing the full controlled potential of our own biological systems.
A powerful thought indeed. Where else could this kind of control over our own biology take us? Something to ponder. Well, thank you for joining us on this deep dive into CAR T-cell therapy. It's a complex but truly fascinating area of science and medicine. Keep that curiosity going.